doing, Jeff? Test. Yep. Okay, if everybody can begin to take their seats and we will get going. Well, thank you everyone for coming tonight. I'm Wayne Carter, the CEO of the Kansas City Area Life Sciences Institute, and it's my pleasure to welcome you today to hear from Nobel Prize winner Dr. Randy Schekman. The Life Sciences Institute functions to grow the life sciences in our region, and we function in 4 key strategic pillars: research, education, collaboration, and commercialization. Research is the foundation of the life sciences. Research obviously is a key foundation on which Randy Schekman has been operating for many years. Research fuels our industries. In the Kansas City region, over the past 3 years, we have grown our Kansas City life sciences industry and added 4,000 new jobs to the Kansas City area. And that is a result of the research, obviously, that is being done today and fueling commercialization not only for today, but obviously for tomorrow and tomorrow's jobs. And so research is absolutely key to what we do across our region. Research also fuels Dr. Schekman's passion, as you will soon hear. His research is a reminder to all of us to explore uncharted areas for the hope and the opportunity of new discoveries, innovation, and new cures. Dr. Schekman is also a pioneer in areas other than cell function. He is leading the charge for open source data and has started a new journal, a new scientific journal called eLife. eLife is a free online journal, a scientific journal, publishing articles ranging from biochemistry to computational biology to neuroscience and plant biology. His work is literally unlocking the potential of data which has been sequestered in scientific journals for decades. As a quote from Dr. Schekman's homepage, eLife editors want early career scientists to thrive, see if I can read this, to receive clear and constructive input on papers, quick decisions, and great exposure. Dr. Schekman, as you can imagine, is also a staunch supporter of collaboration. And as I mentioned, the Kansas City Area Life Sciences Institute, one of our key strategic pillars that we work on across this region is collaboration. Collaboration was also identified in our strategic path to 2025, which we published last year, and Dr. Mark Hahn is chairing our collaboration task force. Soon we will be announcing a new program coming forth in the collaboration space, which is going to be called Collaborate to Cure. And so I ask all of you to stay tuned, as you're going to hear more about that opportunity as we continue to grow opportunities for collaboration in our region. Collaboration is also not new to Dr. Hahn. Last year, KCU, along with UMKC and KUMC, developed the first of its kind three-way collaboration for research in the neuromuscular area. It is my pleasure to introduce at this time Dr. Mark Hahn. He is leading our community, leading the life sciences, and certainly leading the Kansas City University in great ways and new highlights. So, Mark.
Thank you, Dr. Carter, and thank you for your leadership of collaboration and collaborative research in this Kansas City community. Kansas City University is honored to co-host this event tonight. KCALSI has been a leader for years in driving collaborative research for this community, and Kansas City University is proud to co-host Nobel laureate Dr. Randy Schekman this evening. As part of KCU's 100th anniversary celebration and our commitment to excellence in medical and scientific education, and of our new enhanced focus on scientific research and scholarly activity, Dr. Schekman is here to celebrate our research symposium tomorrow on our campus. Since opening our doors 100 years ago, Kansas City University has had a significant impact while staying true to our original mission of improving the well-being of the communities we serve. KCU is now the 12th largest medical school in the nation, the largest medical school in the Midwest, and the second leading producer of physicians for Missouri and for Kansas. Right now, more than 8,000 Kansas City University graduates are practicing medicine and science throughout this country. In 2017, KCU will open a second medical school in Joplin, Missouri. It's the first new medical school in Missouri in 50 years. The focus will be on primary care and prevention. Indeed, tonight we have the distinct honor to have Dr. Randy Schekman join us to share the insights of his work, the impact of basic scientific research, and also the impact of delivering scientific information and how it's shared in the 21st century. Now, to introduce Dr. Schekman, I'd like to call upon our Vice President for Research and Sponsored Programs, Dr. Jeffrey Joyce. Dr. Joyce.
Thank you, Mark. It's going to be my real pleasure to not only welcome you, but in a brief minute, welcome Randy Schekman to the platform to talk. Randy is, as you know, in the Department of Molecular and Cell Biology at the Howard Hughes Medical Institute in the University of California, Berkeley, where he's been there for several decades. If location, location, location is a mantra for real estate, then the parallel in science is the laboratories of our mentors. The lineage for Randy is remarkable in its own right. As he entered high school, his future biology teacher, Jack Hoskins, introduced himself as he stood in front of his project at a county science fair and told him he would be taking care of him for the next 4 years, and cemented his lifelong interest in research. During his undergraduate years at UCLA, he became so immersed in his research in the laboratory of Daniel Edward Kauffman that not only did he decide not to go to medical school, but apparently his academics got a little weak at one point. Somehow he managed to graduate cum laude, and went on to do his doctoral research at the laboratory of Arthur Kornberg at Stanford, who shared the Nobel Prize in Physiology or Medicine in 1959 for his discovery of the mechanisms of the biological synthesis of DNA, perhaps preceding the way Randy would be able to go. Randy had a very short postdoctoral stint at UCSD with John Singer. He tells me he got to think a lot about what he was going to do next, and landed in 1976, the beginning of his career at UC Berkeley, fusing the fields of biochemistry, cell biology, and genetics to identify the molecular mechanisms of the processes of membrane assembly and vesicular trafficking in yeast cells. Strikingly conserved, these trafficking mechanisms are at the heart of neurotransmission, hormone secretion, cholesterol homeostasis, and metabolic regulation, laying the foundation for our understanding of a host of diseases in humans. I have a list of awards, but I think two are obviously very important. In 2002, he got the Lasker Award. As many of you know, is the predecessor to the Nobel in many people's eyes, which he shared in 2013 with his colleagues, James Rothman and Thomas Suttle. As important as his research is, as Wayne pointed out, he's incredibly involved in promoting this open source publication of research. As former member of several editorial boards and as the editor-in-chief of the PNAS, in 2011, he resigned those posts and established the opening and editor of eLife, a new high-profile open access journal. Please welcome me in having Randy come and give us a short presentation this evening, followed by questions. Randy, thank you for coming.
Well, thank you very much, Jeff and Wayne and Mark, for those very generous remarks. I'm delighted to be here. My second visit to Kansas City. I had a whirlwind day today and another one planned for tomorrow. What I'm going to tell you about this evening, though, is not so much my own work, but the importance of basic science in technology and medical advances, something that we all rely on, and which we can be proud of, as America is the premier country in the world in biomedical science. The strength of American biomedical science is rooted in the origins of government support for basic science. Government funding of basic research in the U.S. started after World War II with a transformative report, Science, Endless Frontiers, written by Vannevar Bush, the science advisor to Presidents Roosevelt and Truman. He wrote, "Scientific progress on a broad front results from the free play of free intellects working on subjects of their own choice in the manner dictated by their curiosity for exploration of the unknown. Freedom of inquiry must be preserved under any plan for government support of science." Remarkable words of a visionary leader over 50 years ago. I want to share with you a few examples of the many that any scientist can recount that points to the importance of basic, curiosity-driven, undirected science. My mentor for graduate school, Arthur Kornberg, as you heard earlier, was a renowned biochemist, perhaps the greatest biochemist of the second half of the 20th century. He was grounded as a physician in understanding biological processes, and he sought to understand them by breaking them down into their molecular constituents. In the 1950s, after the discovery of the structure of DNA, Kornberg, then at Washington University in St. Louis, devised a way of breaking bacterial cells open to extract protein molecules to discover an enzymatic activity that was capable of copying a DNA molecule, faithfully stitching nucleotides together to make a complement of DNA. A remarkable achievement, the first such enzyme that was shown to take its directions from a template to faithfully copy DNA. Now, the history of that enzyme goes on. There is much that was learned subsequently by his lab and by many other labs, but no one could have predicted in that and the other examples that I'll share with you, the practical application of that discovery. It was never intended to be anything other than a fundamental exploration of the nature of life. And yet, 30 years later, a discovery was made that that enzyme is capable of copying DNA in a way that can be amplified over and over again to start with as little as a single cell and to produce millions of copies of DNA in a procedure that you've all heard of that's called polymerase chain reaction. It is the basis of modern forensic science and much of molecular biology. It is a billion-dollar industry, that one reaction, polymerase chain reaction. It is entirely based on the discovery of Arthur Kornberg, an amazing investment by the NIH in his science. I learned from Kornberg how to approach a problem and to take it apart piece by piece, molecule by molecule. I learned that in his laboratory, and I applied the logic of his approach in my own work on how protein molecules are packaged for export from a eukaryotic cell, a cell that has a nucleus. Most of the cells in our body produce proteins. Humans produce roughly 23,000 different proteins. They're all manufactured inside the cell, but some fraction of them, maybe 25% of them, are designed to work outside of the cell. You know these molecules. They are molecules like insulin or antibody molecules, the proteins in your blood. They're all manufactured inside of a cell. They have to be encapsulated and then delivered to the cell perimeter and then expelled to the cell exterior. The broad outline of that process was established by a pioneering cell biologist, I think one of the great cell biologists of the 20th century, a man named George Palade. I learned from Palade how this process may work, but in my own laboratory at Berkeley, I devised a way of discovering the genes and ultimately the proteins that execute that pathway by studying this process in a simple organism, baker's yeast, one that had not yet been used to evaluate this process. And we were rewarded with our discoveries of the genes that are required not only in yeast, but also in mammalian cells. The genes that we discovered have their counterparts in our bodies and are involved in the production and secretion of molecules like insulin. They're involved in the communication between nerve cells, in the transmission of neurotransmitters that allow nerve cells to communicate with each other or to talk to muscle cells. The very same genes that operate in yeast operate in the human body, one of many examples of the power of evolution to conserve important functions. Now again, as with Kornberg's work, my work was motivated purely by an interest in fundamental discovery about cellular mechanisms. We were not concerned with how that knowledge may be applied because of the conviction that when you make fundamental discoveries, there will be entrepreneurial people to discover a use for that knowledge. Indeed, in the early 1980s, after we made our discoveries, the biotechnology industry was growing up in the Bay Area, and a company contacted me to help them use our knowledge about yeast to manufacture human proteins of clinical interest. Since yeast cells produce and export proteins the same way human cells do, the logic was that if one simply introduced into a yeast cell the gene for something like human insulin, it may be possible to manufacture and secrete human insulin in large fermentation vats. In fact, now, many years later, 1/3 of the world's supply of human recombinant insulin is made by secretion in yeast, again relying on the fundamental principle of evolutionary conservation. In the same way, this company was able to manufacture the hepatitis B vaccine in yeast by expressing the major protein of the hepatitis virus in yeast. The protein became packaged into little capsules the very capsules that yeast cells use to export their proteins were employed for the purpose of creating these vesicles, these capsules in yeast cells. And if any of you have ever been vaccinated with hepatitis B vaccine, you will have received these little capsules manufactured in yeast cells. 100% of the world's supply is made in yeast. Again, completely unpredictable. Anyone who would have guessed that kind of an application would have been just dreaming about it. I want to give you a few more examples, more recent examples of discoveries that I think are now on the forefront and will, I'm confident, lead in the next few years to Nobel Prizes. Let me share with you one of them, of a former colleague of mine at Berkeley, an immunologist by the name of James Allison, who is now on the faculty of MD Anderson in Houston. Allison was an immunologist interested in understanding how the primary attack cell, the T cell, might be employed to try to strengthen the immune reaction against cancer. He was studying a protein that humans produce, that animals produce, called CTLA-4, a protein of the immune system that holds back, that restrains the action of T cells in attacking our own cells, instead allowing those T cells to attack foreign microorganisms or viruses. So this is a protein that we use to keep T cells from acting on our own tissues, causing an autoimmune reaction. He reasoned that if one could put a temporary halt to the action of this protein, the CTLA-4 protein, that perhaps the T cells would be unleashed to now unrestrained attack cancer cells. He did this work in a very basic experimental setting, not in a medical school at Berkeley, and found that he could inhibit the action of the CTLA-4 protein and cause mice to unleash the attack by T cells on cancers, on tumors, and rid the animal of tumors. Now, there's not much of a market for curing mice of cancer. Unfortunately, there are many ways to cure mice of cancer, but this particular technique he then applied, taking his work to Sloan Kettering. He was able to apply to biotechnology companies, ultimately to pharmaceutical companies, and just several years ago, the procedure was licensed by the FDA and has unleashed, as I think many of you know, a revolution in cancer therapy. Immune therapy is now the forefront for treatment of a variety of tumors. Dramatically, melanomas, which were a death sentence just a few years ago, many of them can now be controlled. The cure rate or permanent remission rate using this immune therapy for melanoma and other forms of cancer has dramatically increased. Another basic discovery that leads to an enormous promise. Let me finally, for an example, leave you with another colleague of mine, still a colleague of mine at Berkeley by the name of Jennifer Doudna, a really very talented biochemist who has studied the structure of RNA and the structure of RNA protein complexes. Doudna, just 4 years ago, collaborated with a French scientist to develop a technique that is now generally referred to as genome editing. I think you've probably heard about this, but let me explain it a little bit. Bacteria have their own immune system. They have a way of keeping invading viruses at bay or keeping other bacteria away from each other. And one path that bacteria have developed is an enzyme called CRISPR. CRISPR-Cas9 is the system that has been exploited by Jennifer that consists of an enzyme, a protein, with an RNA molecule that allows a foreign DNA to be touched and inactivated by chewing it up. Jennifer discovered that she could make a synthetic RNA molecule that would equip the enzyme to identify and hit a gene in any organism, virtually any organism. This was published in 2012, and it has set off an incredible revolution with the promise of treatment of genetic diseases. For example, it may be possible to cure AIDS. There is only one person in the world who has been cured of AIDS, but there is a way of doing it, And that could employ her discovery to inactivate the gene that encodes the receptor for this virus. There are people in the world who are naturally resistant to the disease, to the virus, and all one needs to do is to inactivate that gene, and one can be cured of AIDS, and I think the CRISPR system will allow that to happen. This is a basic science discovery that comes from studying how bacteria work. No one, again, could possibly have imagined that such basic science discovering enzyme, discovering genes in bacteria could lead to this kind of a revolution, and yet it is a revolution indeed that will be worth billions of dollars, and there's a big patent dispute over that right now between MIT and Berkeley. So these are dramatic developments, but they are not unique. The history of science is filled with examples where a fundamental discovery of how the natural world works can be turned to practical advantage by people, I think generally in the private sector, who have an entrepreneurial spirit. That is really one of the great gifts that we have in the U.S., a venture capital system that allows the work in universities and research institutes to be directly translated into science. The Bayh-Dole Act was a visionary act of Congress that made possible a productive relationship between scientists, basic scientists in universities and research institutes, and the private sector. It is the genius of American capitalism. Many of you can recount similar stories where an investment in basic science has resulted in a direct application to medicine and technology. And yet, we find a growing tendency for government to attempt to manage discovery with expansive so-called strategic science initiatives at the expense of the individual creative exercise. Louis Pasteur recognized this tension long ago. He wrote, "There does not exist a category of science to which one can give the name applied science. There are the sciences and the application of science bound together as the fruit of the tree which bears it. American biomedical science will flourish only so long as our elected representatives continue to invest in the creative genius of our finest biomedical scientists." Thank you.
So my primary role will be to bring the microphone around to anybody for a few questions. I'm going to open it up because we had a great discussion earlier today with some of our faculty about the editorship of eLife and the role of open source publications, and Randy's real concern about what he considers luxury journals and suppression of research. If you could give us just a short synopsis of why you think research impact is probably not, by citation, is probably not the best way to go.
Sure. So this is a now more specialized interest of mine that developed beginning about 10 years ago when I was appointed as the editor of the Proceedings of the National Academy of Sciences, a major science journal that is run by the U.S. National Academy of Science, a fine journal. I published many of my most important papers in that journal. But I learned as my term as the editor progressed that increasingly young scientists were driven by pressures of funding and of promotion to publish in the very small, very few, very select journals that are run by commercial interests. Nothing against commercial interests, but the decision-making process at the journals, the so-called high-impact journals, specifically the journals called Nature, Science, and in the life sciences, Cell, the decisions are made on the basis of commercial interests and are done by professional editors who are hired by these journals to make the business decision that results in the sale of magazines. Nothing wrong with that, but it does, I think, distort the nature of the science that young scholars choose to choose to engage in. It causes young scholars to work in areas that may be more popular because these journals are excessively focused on the number of citations that a paper will generate and the immediate buzz that a paper will create, the exposure that one gets in the newspaper or in the press. This is, I believe, antithetical to scholarship. Many of the great discoveries that one can recount were not immediate sensations. They were great discoveries because they were original. They weren't the same things that other people were doing. They were different, and so they take years to be discovered. And yet, the commercial journals, Cell, Nature, and Science, base their choice of papers on a number. Some of you may have heard of it. It's called the Journal Impact Factor. That Journal Impact Factor is a false measurement of scholarship. It measures the influence of a paper, of a journal, over just a 2-year window. And I submit to you that some of the most important discoveries were published and then not recognized for many more than 2 years. So I've been very concerned about this and the influence of that false measurement of scholarship. With the National Academy of Sciences, I took the journal as far as I thought I could take it, but then I was given an opportunity to start with a brand new journal funded by generous support from the Howard Hughes Medical Institute, the Wellcome Trust, and the Max Planck Society. It's an open access journal where all the decisions at every level are made by active scholars, not by professional editors. And so it's scientists in charge of this journal. And this is an effort to try to wrest control of the most important life science literature away from professional editors and return it to, I feel, where it belongs, and that is in the academy. So I've taken very strong positions on this. On the day of the Nobel Prize ceremony in 2013, I published an editorial in The Guardian where I said that I would no longer publish in these, what I refer to as luxury journals. I took a lot of heat for that. I was accused of hypocrisy, having published in those journals in the past. I was complaining about them for years, but it wasn't until 2013 that anyone would listen. So I have continued to speak out wherever I'm given an opportunity, and I'm happy to do it again this evening.
So, are there any questions? I'm happy to bring the microphone to you. I'll talk, boss. I know you're not. Okay.
I was just wondering, Randy, what you think about the pressure at NIH for funding of translational research, and
Yeah.
what has that done to sort of what you have espoused, especially going back to the Secretary under Roosevelt?
Well, it's part of what I refer to as the sort of government attempt to control from top down the management of science. You know, it's the National Institutes of Health. It's not the National Institutes of Basic Science. But it is our responsibility to explain what role basic science plays in biomedical science. And it doesn't help when the leadership of the NIH is excessively driven by management. And so I objected to the creation of, not that anybody listened, but I objected to the creation of the Institute for Translational Medicine at the NIH. I know medical school deans love that sort of thing, but I have to say that biomedical scientists at universities and medical schools are ill-equipped to do drug discovery. That should be done in the private sector, I believe. The private sector is in a better position to make financial decisions about where to invest funds to their benefit in creating drugs that have commercial value. We in the universities are ill-equipped to do so. And so forcing us to be drug discovery laboratories, I think, is a misuse of public funds.
Thank you for a wonderful presentation, Dr. Schekman. Cindy Russell, UMKC, School of Nursing and Health Studies. One of the things that we're facing around the country, particularly being on a P&T committee and reviewing promotion and tenure materials, is this entire degradation of the funding mechanisms for science. Can you talk a little bit about what your projection is for that in the future and how you think that might impact basic science?
Yeah. Well, it's a very serious problem. The NIH budget has grown dramatically in my career, but so has the census of biomedical scientists. It's grown even more dramatically. So there's enormous pressure. I mean, we can't continue to grow exponentially. There's never going to be enough money. On the other hand, I think there could be a better way to invest the money that's available. I think some of the money gets spent on big science. I mean, the current director of the NIH is a proponent of big science, big laboratory science. I prefer the model that most of us have grown up with, which is R01, investigator-initiated science with manageable groups of people that we can communicate with on a regular basis. I think the very large factory laboratories are inefficient, and so I think a lot of that money gets wasted on those big laboratories. But increasingly, I think we have to turn to the private sector for additional help. When I was a beginning faculty member in 1976, there was no private investment in basic science. There was no startup money. When I started at Berkeley, I got that much money from the campus to start my laboratory. It was all the federal government. But now young faculty can expect to get a setup package from their home institution. They can often, if they're really good, they can expect to get private foundation grants. And increasingly, I think we're going to turn to wealthy benefactors to support basic science. There is now an organization called the Science Alliance, which includes some of the most wealthy, far-sighted individuals in the country who are joining forces to encourage young philanthropists to invest their wealth in basic science. And so I think that's an encouraging development that could help.
Hi, Carolyn Henry from the University of Missouri. I was struck in your introduction by the fact that you seem to have been touched very early by some mentorship that may have led you to make the decisions you did in your career, and I wonder if you can speak to how we as scientists can better foster that and how we can reach students at a very young age.
Well, one way is to teach. I love teaching. I teach at Berkeley, I teach a freshman seminar, so these are 18-year-old kids. I teach it on a topic of my own choosing, which is, the title of my class is Insulin as a Window on Discovery in Biology. So I start with the discovery by Banting and Best of insulin. And it's a marvelous way to introduce young students to the magic of biochemistry and cell biology. So I think I think teaching for me is offered to me a dimension that I would not have had in the laboratory. I was a graduate student in a medical school department, so I had very little exposure to teaching. I wasn't even all that enthusiastic about it at first, but it's given me opportunities that I can't have imagined. So I think to all who have a laboratory, I think being exposed to undergraduate students is really crucial.
Thank you so much for your insightful comments. I'm Rebecca Johnson, a professor of veterinary medicine at the University of Missouri-Columbia. And I'd like to hear a little bit more about this whole notion of scientists as entrepreneurs, and the combination of our work, which is in my experience rather challenging, to speak the language and work together with our colleagues in business to make those entrepreneurial ventures happen.
Yeah. Well, again, I mentioned this, and as you know, the federal legislation of some decades ago, the Bayh-Dole Act, encouraged more open interaction between scholars and universities in the private sector. It offers an opportunity for scientists at universities to patent their work and to enjoy some profit from it, and then to share their knowledge with the private sector. And I've seen many colleagues who do that, either by creating companies from their work or by consulting with companies. And for me, having no business experience and really no practical sense almost whatsoever, for me, the opportunity to see my basic science applied, as it was in the biotech sector, was exhilarating. I mean, to go to a company and to see, it's a very different style of work. It's kind of more of a team effort rather than an individual effort. To see a team effort give shape to an idea and to develop a product was a revelation. The company that I consulted for was called Chiron. They were the ones who developed these products that I mentioned, but perhaps the most dramatic thing they did that I was able to watch was their discovery of the hepatitis C virus. Some of you may know this story. In the early 1980s, there was still a persistent source of hepatitis virus infection by what was referred to as the non-A, non-B so-called virus, but it was not, no one knew about it. So it was pretty bad. And this company managed to identify the virus in the blood supply and clone it and sequence it and produce antibodies against it, produce reagents that blood banks could use to screen the virus, and it basically removed the virus from blood donations. I mean, it was a remarkable discovery that any basic scientist would have been proud to do, but it took a team effort in the setting of a company to do it. So for those of you who've had that opportunity to have that kind of a relationship, it really, it's for me, it was a revelation.
I want to thank you, Randy, for coming here this evening. Thank you, everybody, for joining us this evening.